champions, today we are going to decode NCERT. And what in NCERT? The most important chapter of biology, that is principles of inheritance and variation, which could give you a total number of 13 marks in the examination, which is not a small number. And I just want to remind you before getting ahead that what is the purpose of this particular series? The purpose is to save your time and energy. Basically, we are going to highlight the most important portions of the chapter. We are going to tell you what kind of questions could have been framed, or basically could be framed on the day of your exam as well. We are not going to explain the concepts. Please make sure that you are studying the concept with the help of Brahmastra series. Over here is just to save your time, so this video would not be going too long. It will be 20 to 30 minutes or even below that. Okay, so the sole purpose is to save your time and energy. Just focus. I will be just focusing on the things which gives most amount of questions. The rest of the things that I am going to skip means it's not like you do not have to read it at all. You just have to read it once, that's all. Do not invest too much of your time reading those content which I'm not highlighting. Okay, so let's get ahead, my dear children. From here your chapter starts, introduction and all. Just go read it once. Now from here you see Mendel's law of inheritance. Now over here it is mentioned that Gregor Mendel conducted hybridization experiment on garden pea for 7 years. The time span is being mentioned over here. And the question comes, in which year or from which year to which year did Gregor Mendel do his hybridization experiment? You should be knowing it is 1856 to 1863. For 7 years, my dear children. Okay, going ahead. The next question that could be coming is, how many pairs of characters were studied in Mendel's experiment? So there were 7 pairs of character, or we can say 14 true breeding pea plants. 7 pair means 14 plants were there. So you should be knowing the number as well. And these characters were contrasting characters. Now over here there is a small box that is given where they have mentioned the characters which were being studied. Now the next type of question that could come is, which one of these characters are not studied by Mendel in his experiment? So along with this, they can give you one extra thing. For example, leaf shape. So leaf shape is not the character that he studied. So you should be knowing about that. Only if you know these things, these characters, that is stem height, flower color, flower position, pod shape, pod color, seed shape. Seed color were the only characters that he studied. You will be able to answer that as well. I hope I am clear. And sometimes they will be just asking you, tell the name of the characters. Tell the name of all the characters that were studied by Mendel. So please be prepared. Study this particular thing. Okay, going ahead, we have this thing over here. So basically he conducted his experiment. He got F1 generation, the first generation, and then he got F2 generation. Now in F2 generation, my dear children, the plants were looking different. So he took tall plants, he took dwarf plants, fused them. In F1 generation, all of them were tall. All of them were tall. In F2 generation, he found that 3 of them were tall and 1 of them was dwarf. So this ratio that you have, okay, this is phenotypic ratio. You have to remember it. For F2 generation, the phenotypic ratio is 3:1. What is the genotypic ratio, my dear children? We'll come on to that. Okay, we'll come on to that. But here, this is very much important. 3:1, do not forget it. Somehow this is getting, I have written what is the ratio of tall plants found in F1. That is the thing. Okay. In F1, all of them are tall. In F2, 3 of them are tall. So you should be knowing all this thing. Now over here, these things, okay, have not given questions, but you should be knowing these things, that basically Mendel proposed that something was being stably passed down unchanged from parents to offspring through gametes. Okay, what did he call it? He called it factors. He didn't know about genes and all, so he called it factors. Now the different type of genes, the variety of genes that you have, what do you call it? You call them alleles, my dear children. Now you have homozygous condition and heterozygous condition. You should be knowing about that. Homozygous means the alleles are same, that is capital T, or the alleles can be small t, small t. Anyways, it has to be the same. Okay, heterozygous on the other hand, which you will be seeing in the next this thing over here, somewhere over here I studied heterozygous. Where is it? So it takes some time to load at times. Okay, load. Come on, my champion, you have to load yourself up. Okay, cool. So here there is heterozygous term. That means over here the gametes are not same. For example, capital T, small t. Over here, sorry, not gametes, the alleles are not same. Over here capital T, small t is there. Alleles are different. That means it is heterozygous condition. Now you should be knowing over here, they have mentioned about monohybrid cross. So monohybrid cross is where only one particular character is being studied. For example, height. Height is being studied. Okay, now from this height, for you have tall plants, you have dwarf plants. When you fuse them, you get a new type of plants. Okay, basically it has capital T, small t. So it's a hybrid that is being formed over here. So that is a monohybrid cross. So you should be knowing about what is a monohybrid cross. They can ask you to define it. They can ask you to give an example. And now over here you see Punnett square. They give a question that is, what is used to know the probable number of offsprings and for predicting the genotype. So over here this is used. What do you call this? This is known as Punnett square. Who gave Punnett square, my dear children? Reginald C. Punnett. Reginald C. Punnett was the person who gave the Punnett square. Now over here the gametes, okay, the gametes are put on this side, other gametes are put on that side. They are made to fuse. With the help of this, what do you get? You get to know the genotype. What do you mean by genotype? The genetic makeup of an organism. So here capital T, capital fuses, you get capital T, capital T. This is the genotype of this particular plant. So same like that you have to say. So this is the answer for the question, that is, what is used to know the probable number of offsprings and for predicting the genotype. Punnett square by Reginald C. Punnett. That is important. Okay. Now going ahead, what do we have? Dominant and recessive things you should be knowing it, children. Okay, dominant, I hope you know it, that which is why I didn't highlight it. So whenever I say that I am skipping things, please don't think that you do not have to study it. I told you, you just have to read it once for your understanding. I am telling you what could give you more amount of questions. Okay. Now over here they have mentioned the phenotypic ratio of the F2 generation, that is 3:1. That is 3:1. But they have mentioned the genotypic ratio as well, that is what? 1:2:1, which is very much important. 1:2:1 is very much important. You should be knowing that 1 stands for what? 1 stands for capital T, capital T. 2 stands for what? Capital T, small t. 1 stands for what? Small t, small t. So this is the thing. 1:2:1 ratio is basically on the basis of this. This is known as the genotypic ratio. How does their genetic makeup look? That is what they are asked. Phenotypic ratio as in how do they look. Okay. Now going ahead over here, so what is the phenotypic and genotypic ratio of F2 generation in a monohybrid cross? So monohybrid cross will have different, dihybrid cross which we are going to study, that will have different one. So this is important from that. Now going ahead, they ask you questions from test cross. What do you mean by test cross? So you should be knowing, therefore, to determine the genotype. So for example, there's a tall plant. Okay, I do not know the genotype of this particular plant. What can I do? Basically, I can cross with a recessive parent. If I cross with a recessive parent, depending on the organisms which are being produced, whether they are tall or dwarf, if I see dwarf organisms over there, I would be able to understand what is the genotype of the plant that I am looking forward for. For example, this is an example given. Okay. So over here, okay, I want to make sure, okay, a flower is violet in color, a plant is having violet color flowers. Now I want to understand whether it is violet because it has 2 capital W or because it has 1 capital W. Either way it will be violet only, because this will be dominating over the other one, right? So either way it will be capital W only. So basically what can I do? I can fuse it with a recessive parent, and that is exactly what I am doing. So I have capital W, small w, small w. In that case, all of the flowers which would be coming, this is just test cross. This is known as test cross. I am going to fuse this with a recessive parent, and if all of them are the same type, then I get to know that basically this is what? Capital W, that is, it is in a homozygous condition. If this is the case, then I will get to know that basically 2 of them will be white in color and 2 of them will be violet in color. Okay, that means this particular parent that I fused with is not pure breed. It is heterozygous, capital W, small w. So this is what you mean by test cross. So you should be knowing about that as well. Now, they will ask you, okay, what were the 2 laws that were put down after doing the monohybrid cross? So there are 2 laws that were the Gregor-Mendel's law, there are 3 laws. Okay, there are 3 laws, but 2 laws were put after monohybrid cross. One is law of dominance and the other one is what? Law of segregation. What does law of dominance say? You have to know about all this thing. Characters are controlled by discrete units known as factors. Factors, they occur in pair. The one that will be able to show its character is known as dominant. Okay, it will be overshadowing the effect of the recessive one. That is law of dominance, which is why in F1 generation all of them were tall, because there was a capital allele, capital T allele. Okay. Now law of segregation says that alleles not show any blending. That is important. So what happens in law of segregation? Basically, if you have a pair of alleles, only one pair is going to one gamete. Only in a pair, not only one pair. Basically, one in a pair will be going into one gamete. For example, capital T is there. Okay, or basically capital T, small t is there. Okay, in the gamete, for example, egg, either capital T can go or small t can go, because they are on different chromosomes. So the chromosomes are going to segregate. The chromosomes are going to segregate, and only one chromosome is going to go from a pair. Only one chromosome will be going from a pair. So this is law of segregation, which says that though the parent contains two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that gamete receives only one of the two factors. So these two things were put down by Mendel after doing the monohybrid cross, okay, after studying about one particular character. Now going ahead, there were certain things which he didn't expect. Okay, so that was incomplete dominant. So usually we think that one allele is completely dominant over the other one. The question will be asked, on which plant this study was being done. So dog flower, snapdragon, or what do you call it, Antirrhinum species. It is known as Antirrhinum majus, whatever you want to say, you can say. So it is snapdragon, please remember the name of it. Okay, now they will ask you what was found in F1 generation. So in there, when tall and dwarf were fused, all of them were tall. But over here, it was not the case. Over here, it was not the case. Okay, one of them were red. Okay, two of them were pink, and one of them again were white. Okay, so this was not seen. This was not seen initially. So that is because the capital R is not completely dominant over the small r. So you should be knowing about that. So, but the phenotypic ratio has changed from 3:1 dominant ratio. What happened? R was not completely dominant over the small r. So they will ask you to make this also. So be sure that you know about all this thing. P generation, that is parental generation, the gametes, the F1 generation, and when you fuse the F1 generation, the F2 generation, what do you get in the F2 generation as well? So F1, F2, everything is important in incomplete dominance. They can ask you for the definition also. What do you mean by incomplete dominance? That means neither of the alleles are completely dominant over each other. Okay, neither of them are completely dominant over each other, my dear children. Okay, or basically the dominant one is not completely dominant over the other, which is why they show an intermediate character, that is pink color over here. Now going ahead, what is important over here? Codominance is very much important. They can ask you what is codominance, okay, and they can ask you what are multiple alleles. So for the example of multiple alleles, what do we have, children? You have the blood group. You should be knowing that basically over here in blood group there are 3 different alleles, which is why you call them multiple alleles. Okay, they can ask you that what is the genotype of a person who has A blood group? Who has A blood group, what will be the genotype of that particular person? So over here it is given. If it is A blood group, it can be like this, IA. Again, if it is A blood group, where is it? Okay, again if it is A blood group, it can be like this also. So you should be knowing the genotypes. Okay, if it is B blood group, it can be either this or this. If it is O blood group, then it can be either this, no, just this. Okay, if it is AB blood group, it can be this. So you should be knowing the genotypes. You should be knowing what do you mean by multiple alleles. Okay, and why IA and IB alleles are said to be codominant. Why? So that is basically asking you what do you mean by codominance. Here also, basically both of them are equally dominant. Both of them are equally dominant, which is why both of them show their character, that is A and B. Okay, that is what you mean by codominance. Now, okay, going ahead, multiple alleles I had told you. So there are more than 2 alleles governing a character. That is what you mean by multiple alleles. Here also there is an example. You can just have a read of it. Going ahead. Okay, now you are going to talk about dihybrid cross. So you should be knowing the gametes which are there in dihybrid cross, and what do you get in F1 generation, the genotype of the plant. Okay, it is capital R small r, capital Y small y. That is what you get. Now in F2 generation, okay, this is the whole page. This whole page equally is important. This whole page is equally important. Okay, so you have the round yellow parent, you have the wrinkled green parent. What is a gamete being formed? That you should be knowing. From this particular pair, one will be coming. From this particular pair, one will be coming. Similarly, from this one is coming. You are going to fuse. What you get, all of them will be round and yellow, but round and yellow like this. It is not round and yellow like this. Okay, it is heterozygous condition. Now, they have to undergo selfing. So they will be undergoing selfing. Here Punnett square is really important, and what is more important is this, the phenotypic ratio, my dear children, which is 9:3:3:1. They will ask you, okay, they will give you a random number. They will give you a random number and ask you how many of them are basically round green, how many of them are wrinkled yellow. So this is the recombinant, this is the different type of things that you are seeing. In parent, they were round and yellow, and and they were wrinkled and green. So this is a new character that you are being seeing over here. That is what, oh, my hair has come. That is what, round and green and wrinkled and yellow. So this thing you should be knowing. The number, you should be knowing it really well. Cool, works. Okay, and how can you remember? Remember, dominant one has 9, the recessive parent has 1. Okay, now round was with yellow. Let's make round with green now. Wrinkled earth was green, let's make it with yellow. So 3, 3 comes there. So on the basis of dihybrid cross, Mendel put down his next law. What was the next law, children? Law of independent assortment. So according to what is dihybrid cross, you should be knowing that. Dihybrid cross means crosses between plants differing in 2 traits. This time you took the shape of the seed and also the color of the seed, round and yellow, or basically wrinkled and green. These are the shape and the color. When you're studying both together, when you're getting new character there, you call this as a dihybrid cross, my dear children. And after doing that experiment, he put down the law of independent assortment, which states what? When 2 pairs of traits are combined in a hybrid, segregation of one pair of character is independent of other pair of character. That is, for example, round is there. Round is basically a character that is talking about the shape of the seed. Now it can be round yellow, it can be round green. It is not always saying that round will be always with yellow or round will be always linked with green. It's not like that. Why, children? Because a pair of trait, okay, basically what happens in a 2 pairs of trait, one pair of trait will be completely independently assorting from other pair of trait. So from here, from green and yellow, either one will be going. From round and wrinkled, either one will be going. It cannot say that round will always go with yellow or round will always go with green. That is what you mean. So that is dihybrid cross. Again, I am telling you for understanding the concepts. I know at this point of time you might be like, what is ma'am saying? But yes, this is for people who have already probably read the chapter at least once, know about it a bit. Okay. So please do not worry if you don't know the concepts. In Brahmastra series, I have explained everything, like how I explained to a small baby. So please go watch it. If you have not watched it, if you think that you are not understanding the concepts, I don't want you to get, panicky about it. Okay, this is done to make your job really easy. Cool. So that is what is important from here. Just have a quick read. Now, okay, what law was put down after dihybrid cross experiment? What is the phenotypic ratio of F2 generation? That is 9:3:3:1. Now, example, in a dihybrid cross in a pea plants, Mendel got 9:3:3:1 phenotypic ratio. It denotes that. What does it denote? It shows that the law of independent assortment is there, or basically this was an MCQ question. Okay, so it states basically wherever you see this particular thing, that is, from 2 pairs of traits are combined, one pair will be completely independently assorting from the other pair. So this was given in an MCQ. You should be knowing what do you mean by that. So instead of a direct question, they kind of twisted it. Okay, now chromosomal theory of inheritance is there. So who noted that the behavior of chromosomes was parallel to the behavior of genes? This question was there, children. And who was the person who did that? Basically, it was Sutton and Boveri. It was Sutton and Boveri who thought like that. So these paragraphs are really important. First thing, where 3 scientists, De Vries, Correns and Schemark, independently rediscovered Mendel's results on the inheritance of character. These people took Mendel's experiments again back and then they kept on talking about it. They found their own things. Okay, this led to the discovery of structures in the nucleus that appear to be double and divide just before each cell division. These were known as chromosomes. Now there were other people who said that the factors that he was talking about, it has something to do with the chromosomes. Later Sutton and Boveri said that it is actually there. Probably these genes factors that are there, it is on the chromosomes. So whenever I say a gene is going, that means basically a chromosome is going. Okay. So Sutton and Boveri noted that behavior of chromosomes was parallel to the behavior of genes and used chromosome movements. Okay, and it is the important things to remember are that chromosomes as well as genes occur in pair. The 2 alleles of a gene pair are located on homologous sites of homologous chromosome. That is, basically on a pair of chromosome, you will be finding the genes. As simple as that. Now, this chart is there, important, you can read it. Okay, you can be asked about the differences or basically comparison between gene and the chromosome. Now, coming over here, this is important. Okay. Who put down chromosomal theory of inheritance, my dear children? Sutton and Boveri. Okay, so Sutton united the knowledge of segregation with Mendelian principles and called it chromosomal theory of inheritance. Now Thomas Hunt Morgan, okay, Thomas Hunt Morgan was hunting, okay, he was basically wanting to find all the things correctly, okay, he was putting more effort and he was doing more research, and he researched on Drosophila melanogaster, that is fruit fly. And why did he choose Drosophila melanogaster? That is given over here. That is a very important question, so please know that. So what are the conclusions drawn by T.H. Morgan from crossing experiments in Drosophila with respect to linkage? That will be the next part. Okay, so over here, why did he choose Drosophila melanogaster? Because it can be, grown really quick, the lifespan is really small, so which is the reason. So there are few things that are given up over here, read that. Okay, it is very much important. They could be grown on a simple synthetic medium in laboratory. They complete their life cycle in about 2 weeks, and a single meeting could produce large number of progeny flies in points. Write it clearly. Okay, now what did he understand? He understood that basically some genes were tightly linked, okay, and while others were loosely linked. So this is what he understood. So how do we understand that? Basically linkage and recombination was found. That is, some genes, basically they are close to each other. They are very much close to each other. So you call this tightly linked genes. Okay, if in a chromosome one gene is over here, other gene is over here, you call them loosely linked genes. Now one very important thing, what happens is that when chromosomes slide together, they kind of exchange their material. So for example, A and B are very close. So when things are being engaged, both A and B get exchanged. So I don't see any new character. It will be same thing. But suppose A is here, B is here, C is here and D is here. A, B, C, D. So A, B is a combination, C, D is a combination. What is happening if they are far away from each other? This B can move that side. So now, and this C can come here. So it will be A, C and B, D now. So you see there is a different combination of genes that have come in a chromosome. This is what you mean by recombination. You are recombining. There is new things that is being formed. So what do you understand? The genes which are closely packed, they will show less amount of recombination. There won't be any new things. But genes which are loosely packed, okay, they will show high amount of recombination. Cool. So this was given, and over here this is very important, my dear children. Genes white and yellow were tightly linked and shows only 1.3% recombination. So what genes, children? Yellow and white. These two genes, okay, they were tightly linked, and if they are tightly linked, that means they are close. Will they show more amount of recombination? No. So 1.3 is the recombination that this shows. So if the question is asked, if some gene shows 1.3% recombination, is it closely linked or loosely linked? So you have to say it is closely linked. Okay, so know that thing. And while white and miniature wing showed 37.2%, so please remember which all showed 37.2 and which all showed 1.3. So white and miniature showed 37.2% recombination. Again, that means if it's too much amount, 50 is the maximum recombination, 50% is the maximum recombination. 37.2, that means it's a great number. So basically you should be knowing about that also. 37.2, that means it has high amount of recombination, that means this is loosely packed. So 37, this is a question, okay, 37.2 recombinant Drosophila progeny obtained in T.H. Morgan's dye hybrid cross experiment with the phenotypes red eye color, normal body and white eye color miniature is due to what? Because they show loose linkage, okay, because they are not closely linked. Now again, over here we have come to polygenic inheritance. You should be knowing the example of polygenic trait. Example is your skin color. What do you mean by polygeny? Basically different genes are there for coding for one particular character. So you should be knowing about ABC gene. If what happens if capital ABC comes together, what happens if small ABC comes together, you should be knowing about that. Over here pleiotropy is given. Example of what do you mean by pleiotropy? Where a single gene, okay, a single gene is usually for one character, but this single gene is there for multiple character. This is what you mean by pleiotropy, my dear children. Okay, example is phenylketonuria disease is being caused because of that. Because there is one mutation in an enzyme, that is phenylalanine hydroxylase, which is why phenylalanine do not get converted to tyrosine. And when it is happening, okay, tyrosine, it is not getting converted to tyrosine, that is one big problem. Apart from that, in people who have this, basically this particular enzyme is not there, they will be having melanin deficiency as well. Okay, there will be melanin deficiency as well, which controls your skin pigmentation and things like that. So basically it is controlling your skin color, and also there is one more, tyrosine is not being produced, so two characters at the same time. So that is why it is known as pleiotropy. Okay, now Henking, it's about sex determination, my dear children. Henking was the person who basically found out basically in insects and all. Okay, in insects, in insects and all, 50% of them carried something, 50% of them didn't. So he was a major guy, just know about him. Okay, and then you have to talk about different type of sex determination mechanism. So in birds, it is what? XO type of sex determination. It is what? XO type of sex determination. That means there will be one chromosome and there won't be another chromosome. In males there is only one X chromosome. In females they have a pair of X chromosome. Females, they have a pair, in males there is only one. This is known as XO type of sex determination. So you should be knowing about that. Then XY type of sex determination is shown by us, and also my dear little cuties, that is Drosophilus. Okay, Drosophila also basically shows XY type of sex determination. Now there is one more type of sex determination, my dear children, before that. That is male heterogamety. Okay, male heterogamety, that is basically males can have two type of gametes. Okay, either in one gamete it can be X, either in another gamete it can be Y. So basically two type of gametes are being produced by the males. In XY, in our case, males show heterogamety. Males show heterogamety. They produce two type of gametes. Whereas females, they show homogamety. Both of the gametes which are produced by the females are what? XX. So it is female homogamety. Now there are cases where females show heterogamety. That is in birds. That is in birds, my dear children. In birds, what happens? For a bird to be a female, oh my god, for a bird to be a female, basically it should be Z and W. It should be what? Z and W. Unlike the male birds. Unlike the male birds which have ZZ. So either way, the gametes which will be found will be Z or Z. So males are not showing heterogamety, but females here are going to show heterogamety, but because it can be either Z or it can be either W. So that is one thing. Now there is whole different type of sex determination, my dear children. So you should be reading this for female of what say human beings. That is very much important. I think you have already studied it from 10th grade onwards. This is very important. Sex determination in honeybee, haplodiploidy is the type. So what is haplodiploid sex determination mechanism? Mention an animal which exhibits this. So it is honeybee that shows this type of sex determination, my dear children. Haplodiploidy. But the haploid ones will become what? Males. Diploid ones will become females. So you just have a quick read of it. Very much important. Very important. Okay. Now mutation, you should be knowing about point mutation. Example is being asked to here. Classical example of a mutation is sickle cell anemia. It is already given. So just go through it. Okay. Now pedigree analysis, this particular thing is important over here. These things are important. Just have a reference. They will give you a pedigree chart and ask you what type of disease do that particular person has. Okay. So disorders also. There are two type of disorders, okay, Mendelian and chromosomal disorders. So you should be knowing which of them are Mendelian, which of them are basically chromosomal. Mendelian disorders arise when there is a problem in one particular gene. Chromosomal when there is a problem in number of chromosomes and stuff like that, okay. So example of Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. Know the examples, very much important. Over here also they have given. Okay, autosomal dominant trait is this particular disease, autosomal recessive this is a disease. So you should be knowing about that. Color blindness is one very important, okay, thing. It is sex linked recessive disorder, please know that. And this happens, this is because the genes that lead to red green color blindness are on X chromosomes. So males have only one X chromosome and females have two. If males have one X chromosome and if that one X chromosome is defected, they will have this disease. But if females have two X chromosome and only one is affected, the other one will overshadow the effect and it will be normal. That particular female will be a carrier. So always remember in X-linked, in sex-linked disorders, no, the females can be a carriers mostly and the males are the ones which are affected. Okay, females will also be affected if both of the X chromosomes are having a problem. So this is the question, which of the following is not possible in a marriage between carrier woman and normal man? Okay, I have solved it in Brahmastra. Color blind boys, carrier girls, color blind girls, normal boys and girls. So you have to make a cross and let understand. Okay, it's a carrier woman, my dear children. So that means only one X chromosome is affected. Normal man, that is none of them is being affected. None of the chromosomes are being affected. So you have to cross it and then see. You have to basically cross. This is XX where this is affected and then you're crossing it with Y, XY. So make a cross and see what is the answer. Will you be finding which one is not possible is being asked. Which one is not being possible? I can show you. So if you cross this with this, you get XX normal one, normal female is there, okay. XY normal male is there. Carrier female is there. And affected male is there. This is carrier female. Why? Because one is there to normalize it. Okay. Over here there is none. So this is affected male. So you will get affected, colorblind boys. You will get colorblind boys. Okay. You will get carrier girls. You will get carrier girls. You will get colorblind girls. Do you get colorblind girls, my dear children? No. So this becomes the answer. What is not possible? This is not possible. Normal boys and girls. Yes. You have normal boy, normal girl also. So what is not possible is this. So you should be knowing about that as well. Works? Now, going ahead, hemophilia, again sex-linked recessive disease. What happens in hemophilia? You should be knowing about that. There is a clotting enzyme, clotting factors which is getting affected, which is why basically the genes which is responsible for the clotting factor that is getting affected, which is why it is not being produced. So you should be knowing about that. You should be knowing who is the carrier, who is not, and everything. Whatever I have highlighted is very much important. Same like color blindness, this is also very much important. Okay, which enzyme or which protein is affected, you should be knowing about that. Sickle cell anemia, again, what type of disease is it? It is autosome linked recessive disease. Until now it was sex linked. It is autosome linked disease. What is happening over here? What are the genes over here? HbA, HbS. You should be knowing about all the thing. Why does it happen? Sickle cell anemia. Because of substitution of what? Glutamic acid. By what? By valine at the 6th position of beta globin chain. So you should be knowing 6th position of beta globin chain. Which codon? 6th codon. What is happening? GAG. In GAG, the A is being replaced by U. The A is being replaced by U. So which is why glutamic acid, which was being produced, it is not being produced. Now it is valine is being produced, because GUG stands for valine. Now this happens in 6th position of the beta globin chain. All this information are very much important. A patient goes to the doctor worried that a simple cut is taking unusual long time to clot. The doctor identified the patient's condition as what? What is his condition? That a single cut is taking too long time to clot. Basically, you studied in hemophilia that clotting enzyme, the factor that are responsible for clotting, is getting affected. So this is the condition. So these are the type of questions that comes from these things. Okay. Now phenylketonuria, again, what is phenylketonuria? Write the cause and the symptoms of phenylketonuria. So you should be knowing in phenylketonuria, phenylalanine hydroxylase enzyme is not being produced, which is why the conversion has not been happening. If conversion has not been happening, phenylalanine gets accumulated. It gets converted to phenylpyruvic acid, causes mental retardation, everything. Very much important. Thalassemia is there. Again, autosome linked recessive disorder. You should be knowing it. What happens over here? Here, basically in sickle cell anemia, there was a mutation. Okay, there was a single point mutation happening over there, right? GAG was getting converted to GUG. That was the condition. And what was happening? Because of this, the cells were getting in sickle shape. The cells were getting into sickle shape, and they were not able to transport oxygen. They were not able to transport oxygen. Now, that is a qualitative disorder. There's a problem in the quality. There's a problem in the quality. Now over here, I'll say it is a quantitative disorder. That is a question that is being asked. Thalassemia and sickle cell anemia, both of them, they are blood disorders. But how they are different? So over here, basically, there are, in hemoglobin, no, there is alpha chain and there is beta chain. If both the alpha chain are affected, then that's a problem. If the beta chain is affected, that is a problem. If one alpha chain is affected, that becomes a problem, but not a big problem. Okay, if one beta chain is affected, that becomes a problem, but not a big problem. So this is the thing. So depending on what all chains are affected, okay, I can quantify it. So that is thalassemia major, there is thalassemia minor, and things like that. I have done this question in Brahmastra. Go watch it. It is very important video. So few autosome linked recessive gene blood diseases occur in human population. Among them, some are related to qualitative and quantitative problem of synthesizing blood proteins. Explain. So you have to give the difference between sickle cell anemia and this, why it is known as like that. Okay. Now we have come to the next thing. You would be asked about aneuploidy, my dear children. So be sure you know about that. Failure of segregation of chromatids during cell division results in the gain of loss of chromosome. This is known as aneuploidy. Now aneuploidy, when you say gain, you say trisomy. When you say loss, you call it monosomy. So over here it is given, okay, these are the examples of that. So monosomy I have marked, trisomy I have marked. Polyploidy is different from aneuploidy. Okay, in aneuploidy what is happening, my dear children? One chromosome might be extra, other chromosome might be lost. That is the thing. Okay, but in polyploidy, what happened, whole set of chromosomes are coming together in one organism, or probably whole set of chromosome might not be there. This is known as polyploidy. Okay, failure of cytokinesis after telophase stage, that is totally different. It is found mostly in plants. Now in aneuploidy, I told you monosomy and trisomy is there. So this is an example of trisomy, Down syndrome. What happens in Down syndrome? You should be knowing that the cause of this genetic disorder, presence of an additional copy of what? Chromosome 21, trisomy of 21. They will ask you the karyotype. Please know about it. Examples also, what do you call, symptoms also. So write two symptoms of Down syndrome and write the chromosome number in Down syndrome. The chromosome number of Down syndrome, what will be the case? Basically one extra chromosome is there. So usually people have 46 number of chromosome, but they will be having 47 number of chromosome. Okay, 45 autosomes. Usually we have 44 autosomes and 2 of them have sex chromosomes. 44 plus 45, 46, right? So that is the usual case. But this time autosome, 21st chromosome is an autosome. So they will be having 45 autosomes and there will be X and Y, or basically X and X. So you should be knowing about all these small details. Okay, Klinefelter syndrome. Okay, mention the karyotype of Klinefelter syndrome. This is the karyotype. Where is the karyotype? 45 XXY. 47 XXY. What happens in them? What is gynecomastia? All this thing you should be knowing. Similarly for Turner syndrome also. So they give you 1 mark question on the basis of this. Okay, for each, one for each. If you're missing out, you're missing out on a lot of things. So these are the things that you should be definitely knowing about this particular chapter. You should not be missing these things, and then please read the summary. It will help you understand a lot of things. I hope that this particular video was useful for you. I don't want to go any further because I think I have taken 4 extra minutes. But I think children, I don't know if you would find it very useful, but actually it is very much important for you to put special attention on these things, especially while you are revising. I'm not suggesting this for a person who is just going to start off a chapter or things like that. For a person who has just started off a chapter, go watch one-shot videos, go watch this particular thing, Brahmastra series. It will be more important for you. But people who are revising can revise these many things, okay, in a fast and efficient way. I don't want your revision to stay longer. So that's all, my dear little champions. If you think that this particular video was useful, do not forget to like, share and subscribe, and do not forget to show us your love because we need it very much. So that's all. Bye-bye and take care.